Hey, hi everyone. It's my great pleasure to introduce Rob Knight. As you can see from his titles, Rob is a very unique academic. He's the Wolf Endowed Chair of Microbiome Research at Brady's Children's Hospital, and he's a professor of pediatrics, bioengineering, and computer science, a combination that we don't often see. Rob was an HHMI early career scientist. He's a fellow of the American Association for the Advancement of Science and the American Academy of Microbiology. He was awarded in 2019 the NIH Director's Pioneer Award for his microbiome research, and he received the 2017 Mastery Prize. He's written books and he gave a TED Talk in 2014 on his research, and his lab has pioneered many of the software tools and laboratory techniques that enabled high throughput microbiome sequence, having a huge impact on the community. He's the co-founder of the Earth Microbiome Project, the American Gut Project, and the company, I'm not sure I'm pronouncing this correctly, Biota. And his work has linked microbiome to a range of health conditions, including obesity and inflammatory bowel disease. And he has enhanced our understanding of microbes in the environment and produced very meaningful data science tools. So, please welcome Rob.
Sorry to interrupt. I don't know if you can hear me, but the second room is making noise.
Congratulations on the Info Center being funded. It's really going to have a transformational impact. And thank you for inviting me to speak here. And I'm glad you were able to fit this in despite the many scheduling headaches. So thank you. What I'm going to tell you about today is some of the bioinformatics and data science issues that are really important in microbiome research, and how you can get involved, and how there's a lot of open problems. And I was delighted to be introduced by Gal, because I'll be highlighting some of the work that our joint student George Armstrong did on dimensionality reduction during this talk. But before I try to convince you that you can get involved in some of the technical issues, I should first motivate you about why you might want to get involved, and what is this microbiome research stuff all about? Why should NSF be supporting it as well as other agencies, and what's going on at UCSD specifically? So our world, it turns out, is very much a microbial world. And microbes are literally the reason why the land is green and why the sky is blue, right? The oxygen in the atmosphere that produces the Rayleigh scattering is mostly produced by microbes. And in fact, there are 100 million times as many bacteria on Earth as there are stars in the universe. And the vast majority of these bacteria are unknown. We're just beginning to find out about them. Microbes are also intimately involved in many of Earth's problems, including a lot of major. I'm sorry, does that need to go higher? Is that better? How about that? All right, microbes are also intimately involved in many of Earth's problems, including a lot of priority areas for NSF and for NIH. For example, in both animals, including humans and plants, microbes are involved in infectious diseases, obviously, but also chronic diseases. We're finding out a lot about their impact on cancer, on Alzheimer's, on other things in the relation to the gut-brain axis. They're involved in food production, they're involved in pollution and biogeochemical cycling, and they're involved in climate change. And so harnessing microbes to address these problems could be really important for humanity and for the rest of the planet. But what they require is better data and better quantitative approaches to dealing with that data, which is mostly what I'll focus on today. So I direct the Center for Microbiome Innovation here at UC San Diego. This is our leadership team of faculty drawn from across campus. Here's our full-time staff. And I'll admit that for the last 3 years, we've been largely focused on pandemic response here on the UCSD campus. So you might have seen the sewage auto samplers or the vending machines for COVID tests and other stuff that we helped with setup of. But when we're not doing that, what we're doing is integrating researchers to address large, complex biological science challenges. And so we have over 150 faculty members in their labs involved across campus already, and we'll be doing another membership drive in the near future. And this just gives you an idea of the breadth of different systems that CMI members tackle. everything from the oceans to pets to studies of individual microbes to food. And here are some of the corporate sponsors that we have who are interested in these topics, which reinforces how microbiome research isn't just in the lab anymore, it's increasingly out there in society. I won't go through all this, but basically we have a highly productive group of members of the Center for Microbiome Innovation, over $25 million in industry sponsorships and directed projects, over $450 million in US government funding. And we do a lot of algorithm development. I'm only going to talk about a few of these tools, but to a large extent, a lot of the underpinnings of microbiome research is developed right here at UCSD by faculty members who are members of the Center for Microbiome Innovation. So in addition to changing how we think about the planet, microbiome research is also changing how we think of ourselves as human beings. And I'd like you to take a moment to consider what you saw when you looked in the mirror this morning and you were getting ready to come to work. For myself, I saw an organism that's just 43% human, and not just because it was early and I hadn't had my coffee yet, but when we think of what makes up our bodies, we've got 30 trillion human cells, but we've got 39 trillion microbial cells, mostly gut bacteria. And that's where that 43% human number comes from, just counting the cells and asking what genome they've got. But you might think it's the 21st century, shouldn't we be thinking about this at the genome level in our DNA? And when we think about that, it's even more amazing, because to our 20,000 human genes, we have a gene catalog of somewhere between 2 and 20 million microbial genes. And so by that measure, counting up the unique genes, you could say we're just 1% human. And what's most amazing is that the 99% of the genes we ignore when we focus just on the human genome are the ones that we can change, that we could take control of through our lifetime and improve health outcomes. And so you can think of that human side of ourselves as basically being like the conductor of a band, where we're really marching along to this microbial drumbeat, where we're starting to understand more and more how those microbes affect our phenotype, affect our health, even affect our mood. And the NIH invested a lot of money, $173 million in the first phase of the Human Microbiome Project, which was launched in 2007, and that my lab was involved in a number of different capacities, where the goal was just to understand what's there in the body, what are the human body's microbial communities and different body sites. And so so the initial papers focused on about 250 people, sampled at up to 18 sites in the body, which, as you can imagine, is a lot of places to stick a Q-tip. And up to 3 time points. And so this resulted in 4.5 trillion bases of sequence data, which in 2012 was a massive data set, but it's what we can produce every single day on the NovaSeq instrument here today. And the key findings from the HMP were about the diversity of the microbes on the human body, the massive variation between individuals. So you're probably 99.99% identical in your human genome to the person you're sitting next to, but you're probably 90% different in your microbes. So each of us has a unique microbial fingerprint. We're starting to use that for all kinds of things like forensics. There's functional overlap where different microbes can do the same job, and we really need to understand that much better. And then there's disease associations, as I mentioned. So the gut microbiome contains all this data that reads out your health, including things like its role in digestion, in the immune system, in brain development, which has just been recently discovered, even protecting against pathogens. And I'll say more about that later in the talk. But it also reads out a lot of things about diseases, including things like chronic kidney disease, other metabolic disorders like diabetes, obesity, and so on, GI disorders, and cancer, which we can now diagnose from a sample of the blood, where we see DNA that's shed from the microbes in the tumor. And the microbiome is also a very sensitive data recorder of all kinds of things that happened to you in the past, like how you were born, how you were fed both early in life and later, what medications you take, how healthy you are in various respects, where you live, and various medical conditions you have. So that's why you should care about the microbiome, whether you're interested in environmental applications, industrial applications, or interested in the human body. So I'm going to say just a little bit about how we acquire the microbiome data and what the structure of it is. There's a whole lot of these technologies that are collectively referred to as omics, which is what we use to unveil the microbiome secrets. So 16S ribosomal RNA profiling is where we read one marker gene where we can basically read out the name tag of what the bacteria are, put them on a phylogenetic tree to understand them, and understand who's there. Shotgun metagenomics reads out fragments of the whole genome, so we can understand at a functional level as well as the taxonomic level what they're doing, as well as who they are. With transcriptomics, we read out the RNA instead of the DNA, so we can read out what genes are being activated right now. And then with proteomics, which is a different technology based on mass spec rather than on sequencing, we can read out all the proteins and their current levels. And then also with mass spectrometry, where you're essentially weighing a whole lot of molecules very precisely, and then smashing them into pieces and weighing the pieces really precisely, trying to puzzle them back together. With metabolomics, what we're looking at is the chemistry of a particular environment, say the gut or your mouth or the soil or whatever, including all the chemicals produced by microbes. So essentially the pipeline is that you get your sample, whatever it is. And what I'm going to talk about today, it's mostly about DNA sequencing. And then you get the microbial abundances, where you're going to get some table where for each sample, for each kind of feature, you have the count of that feature in each sample. So pretty simple data structure. And my lab's been doing a lot over the years to increase the rate at which we can acquire this data. So back in 2007, published in 2008, we did an experiment that collected as many of those 16S ribosomal RNA sequences as all the ones that had been collected by the entire scientific community and released in GenBank up until that point. We did it overnight. And what we did is we took hundreds of samples from the lungs of people with cystic fibrosis, rivers across North America, air samples, incredibly challenging low biomass environment to sample, newly relevant with SARS-CoV-2, and then the Guerrero Negro microbial mats, the most complex microbiome that's known on Earth, which ironically NASA was studying as a model of simplicity until they sequenced it and found out how complicated it was. And so we introduced formal error-correcting tags encoded in the DNA on the sequences from each sample, in a procedure that's now become standard, and sequence them together and unifrac clustering by sample type, which I'll explain more later. You can see that each sample type clusters together by color here, and then you can get more detailed information about what's going on within each of those categories. Now, the most amazing thing is that this would have cost $4 million to do the sequencing if we had done it the old-fashioned way, which cost $8 a sequence. With the then new 454 technology, we did it overnight for just $12,000 instead of over the course of many years. And so this is what enabled the Earth Microbiome Project, the American Gut Project, the Human Microbiome Project, all these large-scale initiatives that look at huge numbers of samples. But what's perhaps most amazing is that that amount of sequencing, 500,000 sequences, that cost $4 million in 2007, 12,000 in 2008. If you do it today across campus at IGM, just a short walk from where you're sitting, it costs you just 75 cents, right? And so there's not many samples you'd sequence if it's going to cost you $4 million. But if it's less than $1, you can think of all kinds of applications where you're going to do spatial sampling, you're going to do temporal sampling, you're going to do large populations, and really figure out how to do it in a data-rich way instead of a data-limited way. So the canonical steps of the workflow for nucleic acid-based sequencing, you get your raw sequence data, you have to do some pre-processing to throw away technical artifacts, which gives you your usable sequences. Then you assign them to features by matching against reference databases typically, which gives you that feature table that I told you about, where for each sample, for each feature, you have a count of that feature. And then you do diversity analyses, which I'll talk about more in a moment. You typically do phylogeny, and then you do various downstream statistical methods, which I'll also say more about in a bit. And so the problem with all of this is how to handle diversity, because every single sequence, every single sample now yields thousands to millions of sequences, depending on what technology you use. We can sequence hundreds of samples at once. We've got to compare all those samples to each other and understand what aspects of the similarities and differences between samples are meaningful. So this is just a bar graph showing you the dominant taxa in a few hundred samples from a study that we did back in 2007. And you can tell that squinting at the bar graph is not going to get you very far in understanding what's going on there. So what we have to do instead is to turn to techniques that ecologists have been developing over the past century and a half. And so the key concepts of ecological diversity have been around for a while, were originated in plant ecology. where what we typically care about is alpha diversity, where you care about diversity within one community. Beta diversity is the differences between different communities. And that's what we're concerned about if we're trying to link a microbiome to health or something like that. We want to know how are sick people and healthy people different from each other in ways we can detect. And then there's gamma diversity, which is at the landscape scale, which is used less. But these concepts underpin most microbiome studies today. We're trying to measure what's in a community. We're trying to measure differences between communities and then relate it to somewhere between hundreds and millions of samples. So the first major issue we hit is how do we measure and integrate community dissimilarity? And so here's a representation of the organism by sample tables, where basically you have relative abundance of each organism in each sample. And you want some function of the first sample and the second sample that takes all of this data and relates the samples to each other into a distance matrix. So if we pick sample B and sample C, the distance from B to C is something that you want to measure and boil down to one number. But the problem is that ecologists have been very creative, and there's not one way to do this. There's a whole lot of different ways to do this. And this gives you just a table from a paper that was published about a decade ago. There's something like 80 different measures that we implement in our chain pipeline at the moment. So one measure that we found particularly effective is one that Kathy Lozapone, one of my first grad students when I was at Boulder, introduced as the core of her thesis, which is called UNIFRAC. And what we do in UNIFRAC is we exploit evolution as a metric for comparing these communities. So the intuition here is if you have two identical communities labeled here red and blue at the tips of a phylogenetic tree, where the internal nodes are common ancestors, and the further back you go, the further back you're going in time. The idea is if you have identical communities, everything you see in the red community, you see in the blue as well. So all the branches on the tree lead to both communities and they're shared. None of them are unique, and we define that distance as 0. Whereas in contrast, if we have two completely unrelated communities, all the branches lead only to the red community or only to the blue community. None of them are shared. We define that distance as 1. Then any actual pair of communities, you'll have some unique branches and some shared branches. You calculate the fraction that's unique. That's the UNIFRAC distance, and that gives you that distance metric. And there's various elaborations on that. So you might wonder, hey, what about how many times this for each of those sequences? So with weighted UNIFRAC, what we do is we weight each edge according to the number of sequences that came from that particular type of community, and then we look at the absolute value of the difference in weights between the first community and the second community along each edge. So we proved the metric properties of both the weighted and the unweighted UNIFRAC metrics in collaboration with Manuel Yedza at Boulder back in 2010. And the reason why it's so uniquely effective is basically revealed when you take the next step, doing principal coordinates analysis on each pairwise distance. So what you can do is you can take Darwin's insight that there's one tree of life that relates all organisms and measure each pair of communities on the tree. So measure red to yellow, measure red to blue, measure yellow to blue, store all those distances in a distance matrix, and then do things like principal coordinates reduction, which I'll get into next, or hierarchical clustering, which I showed you in that picture before. So what's principal coordinates analysis? Essentially, it's a way of taking really complex things and making some sense out of them. So you might get, so when you build your correlation network and, build your distance matrix and turn it into a point cloud, you're likely to get some structure that's very difficult to see anything in. And what you really need to do is find some way of illuminating what's going on in that structure so that it makes some pattern that's more interpretable as a projection. And so with principal coordinates analysis, what we're doing is we're doing eigen decomposition to take the point cloud and then spread it out as much as possible along the eigenvector associated with decreasing eigenvalues. So we're going from a distance matrix to a small number of coordinates that give us a location in space. And so essentially, what we're doing is projecting low dimensional shadows of a high dimensional point cloud. And so principal coordinates analysis is basically telling you where to put the light bulb and also where to put the wall to get a meaningful picture out of what would otherwise just be a mess. So then the question is, what metric should you select? And when you use UniFrac to look at different clusters, so here we're looking at things found in the guts of animals versus things free living in the environment, and you can see they form two nice biologically plausible clusters. But if you do it with a whole range of other distance metrics, ranging from Euclidean to binary dice coefficients and Jacobi coefficients, you see you get these spike artifacts at right angles to each other. And that's largely due to the sparsity of the data matrix. And the reason why UniFrac works so well is that it's using an auxiliary data structure, the phylogenetic tree, where what it's doing is instead of treating all organisms as equally related to one another, what it's taking into account is the structure of which ones are more or less similar according to evolution. And that's why we get, that's how we can reduce the sparsity overall and get biologically meaningful patterns out of the data that are lost when we use a non-phylogenetic metric. So, so then there's the question about how do you do the dimensionality reduction? And in collaboration with Gal, with a very talented student, George Armstrong, in the BISBE program, George looked at how PICO-A and UMAP and also TSNE, although I won't talk about that here, compare on microbiome data specifically, because TSNE is very popular in the single cell field. UMAP's been more recently introduced, but there wasn't a lot of application of either of those to microbiome data. So basically the idea was to take the feature tables that I showed you, turn them into distance matrices, and then use different dimensionality reduction techniques to see how clear the clustering was. And the data set we used was kind of fun. This is from our first study that was originally a biogeography study, but it turned into a forensic study. So initially we were interested in what is the biogeography of the keyboard. Where do the microbes live? Are they spatially clustered on the keys? Does the spacebar have more microbes on it just because it's larger than the other keys, which you'd expect from, say, the MacArthur-Wilson theory of island biogeography, if you're familiar with that. But as a control, we also looked at all the fingertips of the 3 people whose keyboards we sampled. And what we saw when we saw the data is we got very clear clusters where all the fingertips and all the keys of each person clustered together. And then 2 of these people share an office, so we also saw that structure recaptured. So it turns out the subsequent research has shown that we're all exchanging microbes all the time with people we live with or we work with. So you're all growing microbially closer to one another and to me right now, just by sharing the space with each other. But anyway, what you can see is that traditional metrics like HSN, HSN is a compositionally informed metric, and I'll say a little bit more about that soon. But when you run it through principal coordinates analysis, you get a lot of mess in the clustering. Whereas if you run it through UMAP with the neighbors parameter set at 15, so basically localized clustering, you get 3 very nice clusters, and you also get the result that the keys and the fingertips within each person separate from each other, which is a structure that we could not see nearly as easily in the PICO-A. We also wanted to check that the placement of clusters was going to be similar both in high dimensions and low dimensions, which is often a failing of dimensionality reduction techniques. We also wanted to check that we were going to preserve the cluster relationships. Essentially, we wanted to make sure that the 2 people who shared an office were in general going to look more similar to one another across different parameter settings for UMAP, as opposed to principal coordinates analysis, which doesn't take parameter settings and is always the same. Then George introduced this idea of using LDA to quantify the separation between hosts, which worked very well. So basically, the idea is that if you have good separation, you should be able to use LDA to fit linear regressions that separate them more or less nicely, depending on the clustering. And so we're looking both at the ability to separate the hosts from one another, which is an easy task, and the ability to separate samples within each host from the fingertips versus the keys, which is a much harder classification task. And so what we saw here was that relative to PICO-A, UMAP was very good for increasing the same host sample type separation relative to PICO-A. So we think it has substantial advantages there. Now, that's not to say that we think UMAP is necessarily the be-all and end-all dimensionality reduction techniques. And we know that a lot of people in HDSI and one of the focus areas of InCore is to look at additional ways of measuring distance and doing dimensionality reduction. We're certainly very interested to work with you on that as those discoveries accumulate. Okay, so the second major issue I'm going to talk about is how to deal with compositionality. And I think you probably all know about issues with compositional data, so I'm going to go through this pretty fast. But if I'm going too fast, tell me to slow down and I'll go back through it. So I told you that we were getting the count of each microbe in each sample, but that's not really true because what we get back is sequences, and the number of sequences we get back from each sample is a technical artifact that's not linked to the actual abundance in the sample. So what we have to do is we have to do a sum normalization on the sequences from each sample. So if you could actually count what was in each sample, if you're looking at the ratio of what's in sample A to sample B, you know, if the ratio of what's in sample A and sample B is 1, you know that the number of the microbes of that type in the first sample is the same as in the second sample. However, and then similarly, if the ratio of A to B is more than 1, you know that there's more of A than B in your sample. And if it's less than 1, you know that you have less A than B in your sample. However, so also what it means is that if you, so the problem is when you do this conversion to percentages, so when you do sequencing, what happens is that you're converting from absolute abundance to relative abundance because you cannot control the number of sequences you get back from each sample. That's an artifact, it's not data. So as a result, the ratio of the percentage of A to the percentage of B, if it's less than 1, it doesn't necessarily mean that you have less A than B in your sample. What it might mean, for example, so what it might mean is that both of them expanded, but one of them expanded more than the other did, or both of them contracted, and one of them contracted more than the other did. You can't distinguish those situations from each other with compositional data because they lie in a simplex, not in Euclidean space. So the solution to this, which was introduced by Cameron Martino, who's another recent graduate from my lab in the Bisbee program, and Jamie Morton, who was a recent graduate from my lab in CSE, was to use reference frames, much like you would use in, say, astronomy to figure out, you know, are we at rest here, or are we actually going at 67,000 miles per hour around the sun? Depends what reference frame you look at it. And so the extension of this to chemistry is looking at the mass of each solute, like in terms of grams, to the volume of the solution, and then turning that into a concentration. So, Jamie and Cameron really led this with additional contributions from Lisa and Alex, who we collaborated with. And so what reference frames can do is they can cancel out the bias through microbial load, because essentially when you're looking at when you're looking at a ratio of two proportions, each of those contains within it the true number of cells of A and the true number of cells of B that you don't know, and they cancel out. And so by looking at the ratio of two relative abundances, you eliminate the problems of trying to analyze a single relative abundance. So to do this, Lisa performed some really laborious experiments where she could quantify the actual microbial load in each sample with flow cytometry, which is basically a fancy way of counting cells one at a time. And it's exquisitely painful. But that's what got us a relative abundance table and a table normalized for microbial load. And so what we could look at then was which taxa had changed in that relative abundance table. And so you can see that the results after that normalization are near perfect, whereas before the normalization, you just get a horrible scatter that's no better than chance. And so what we were able to show was that using, so Cameron was able to show using the reference frame formulation, introducing the robust Aitchison distance, he was able to massively increase the F statistic and also the k-nearest neighbors classification accuracy using the robust Aitchison method over things like UniFrac, which is phylogenetic, as I showed you, and over Bray-Curtis, which is a non-phylogenetic method. And this works in a range of different sample types. So, so another problem that Cameron was also able to address with this was repeated measures. So the big issue here is that each person has a relatively stable microbiome over time. We also see that in different ecological sites. We even see it with those auto samples that are over campus for the wastewater program for SARS-CoV-2. Each of those manholes has its unique microbiome identity that stays the same over time. And what we're looking at here is an IBD data set where here it's colored by person, and you can see that the different people form nice different clusters of different colors. The age signal is really disrupted by that per person clustering, and you can't see any association of active versus quiescent disease at all because it's just lost in that per individual effect. So, so Cameron came up with this technique called context-aware, called compositional tensor factorization, which is basically a dimensionality reduction method that allows you to see a whole lot of cool things in these time series studies that are confounded by individual subjects. And so basically the idea is you take each of the subjects, each of the time points, and then each of the features, and then combine those into a tensor, and then factorize that tensor so that you can separate out the contributions of subject, of feature, and of time. What's exciting about this is that it can give you trajectories in the principal components of the vectors. So, for example, on that data set I showed you before, where you couldn't see any signature of disease in the PCOA, now you can see a clear signature of the Crohn's cases versus the controls. And what you can then do is you can take it a step further and do feature selection to look at the specific taxa that are associated with either having the disease or not having the disease, and then demonstrate that those are that ratio is several log units different between the Crohn's cases and the Crohn's controls, thus letting you separate them out by phenotype. Cameron was also able to show, and I'll just summarize this in the interest of time, but he was able to show that, that very early life effects shape the microbial community, not just in the first couple of years of life, but ongoing in two different cohorts, the ECAM cohort going up to 1 year of life, then the diabetes cohort that goes up to 3 years of life. And so CTF allows dramatically better discrimination of delivery mode if they're trying to separate out the babies born by C-section from the ones delivered vaginally, even out to 2 or 3 years of age. You can see that the F statistic is dramatically better separating them with CTF than it is with other kinds of distance metrics, even including unified. So again, what you're able to do is to go back and pull out the specific microbial features that are associated with these different states. So Cameron was able to build this microbial birth mode signature, separating out the C-section babies from the vaginal babies, and then validating that it was consistent between these two cohorts. And so here's the taxa that are especially associated with C-section, a lot of which are associated with skin or with the mouth. Versus taxa that are associated with vaginal birth, most of which are more familiar gut taxa that we would expect to see there. And so this microbial birth mode signature was persistent over the first 3 years in both of these cohorts. What's interesting is we could then go to the American gut population and show that in that population you see the same signature over the first 3 years of life, but then in older adults you don't see it, at least in healthy individuals. So we thought, aha, maybe we're seeing this early life signature that then gets erased. But then Cameron looked at the people in the American gut cohort who had inflammatory bowel disease specifically. And if you have this disease, then there's a specific difference depending on whether you're vaginally delivered or whether you were delivered by C-section. Where this is different if you have developed this autoimmune disease, but not if you don't develop the disease. And so what we're starting to see is these early life signatures that are carried all the way into adulthood. We've also worked with Maria Gloria Dominguez-Bello on seeing whether you can restore this maternal inheritance if you were delivered by C-section. And so the study design for this was to take 3 groups of babies, those being delivered by C-section, those being delivered vaginally, and then those being delivered by C-section who were immediately inoculated with their mother's vaginal microbes afterwards. And what was cool about this is that we see that the C-section is different both from the vaginal birth and from the seeded birth in the gut microbiome, in the skin microbiome, and in the oral microbiome. So we're seeing this treatment at birth that has persistent effects in microbiomes over the whole body, potentially allowing us to reverse the connection between C-section and a whole lot of these different diseases. Now, again, this technique is very exciting and has let us go a long way, but I think there's probably a lot more we can do in terms of trajectory clustering, in terms of learning the features that allow us to carry these differences through from birth into later life, and being able to look at how the network structure of the microbiome and the associations in it change from early life onwards. And so having dynamic models that allow us to see those connections and see the connections changing rather than a static network of microbial interactions is another really exciting direction here. Okay, so the third major issue that I'll talk about is applying AI to microbiome data. And the big problem with applying AI and machine learning techniques to microbiome data is that it tends not to be AI ready, in the sense that there's batch effects, there's metadata problems, there's all kinds of other issues that you have to deal with before you can feed it into an algorithm and expect that algorithm to actually work. So increasingly in trying to grapple with this microbial side of ourselves, we find that we run into limitations with human intelligence, and so we have to turn to machine learning and artificial intelligence instead. And towards this goal, back in 2017, UCSD and IBM set up this Artificial Intelligence for Healthy Living Center that I co-direct, which is part of the AI Horizons network, where we're applying initially IBM's Watson technology, but more recently things like Auto AI to analyze the microbiome data. So some of the microbiome patterns, including some of the ones I've showed you already, are pretty simple, and even our puny human brains can decipher them. So if we look at those development trajectories, this is the first large-scale study we did on human development, and what you're looking at is kids from 3 different continents colored in 3 colors, and you can see that all of them approach the adult state by the time they're age 3. And it's very consistent across cohorts, and you can see that essentially all the change happened by 2.5 years, and it's an obvious, straightforward pattern, right? So that's something you can figure out yourself. But it also leads to that alarming question: is your microbiome doomed by the time you're 3 years old? Because if most of the change happens in the first 3 years of life, can you continue to modify it and control it after that in order to benefit your health? So to get at this question in older adults, we turn to the American Gut Cohort, our citizen science project, where we've looked at tens of thousands of samples from people in 37 countries now. And the microbiome changes with age in adults are there, but they're much more subtle. So our first attempt at this, we just used polynomial regression, and you can see the scatter around the points. And I promise you that these regression lines are statistically significant, but they're just not very impressive, right? They're not explaining much of the variation in the data. And what the signal is, is pretty baffling. So this is where we turn to AI and working with IBM to try to figure it out. And so what we did, our initial attempt at this was with random forest regression, where we were able to show that from your stool sample, with random forest regression, we get a much tighter fit, and we can get your microbiome aged within a dozen years from your stool sample. From your oral sample, we can get it to within 5 years. And then from your skin sample, we can get it within just 4 years. So we can get these rather tight readouts of your chronological age from the total microbiome by using more advanced regression techniques. So that leads to the question of how does your microbiome age compare to normal? And the idea is that you might have a progression from young to old, where your microbiome is changing in this clock-like way like I showed you. And then so the question is, if you have a microbiome that's very young, but for your chronological age, maybe you're going to be extra healthy. In contrast, if you have a microbiome that's very old for your chronological age, maybe you're going to be especially unhealthy and encounter health problems sooner. And so we've been exploring this in the FINRIS 2002 cohort. What's amazing about this cohort is it's people who pooped in a cup for science over 20 years ago, and we sequenced all their microbiomes a few years ago, and we have all their health records up to the present. So, so what we can tell is not just your current state and how that relates to the microbiome, but start to predict your future state and ask, is your microbiome healthy, not just in the sense of it's fine right now, but will it continue to be healthy into the future? So a lot of what I've shown you has been done with 16S. A really important problem is how to extend this to shotgun metagenomics, which is the technique I told you about, where we get fragments of all the DNA. This has largely been driven by Daniel McDonald and the Green Genes 2 project, which is about to be published in Nature Biotech. And the problem with green genes is it's built on a reference database with short fragments of sequences, but we'd like to use the whole genome. In the last release we did, there were about 15,000 genomes available. Just a decade later, there were 300,000 available. So we had to deal with a much larger whole genome phylogeny problem. And so essentially, we teamed up with Siavash Mirarab over in ECE to build a backbone phylogeny of all the complete genomes, and then insert shorter and shorter fragments that were taxonomically informative. And you can see that these green patches are places where we only have the short ribosomal RNA tag. We don't have a complete genome. So there's still massive parts of the tree that we still need to discover and fill in, which means that we'll need better phylogenetic inference techniques. So anyone who's tried to take these different technologies of metagenomic data and analyze them together has probably seen something very depressing like this, where you get perfect clustering, but it's by how you did it, not by what the biological samples are. And by putting them on the same reference database, we completely erase that, and the same sample shows up in the same place, whether you did it with whole genome shotgun sequencing or whether you did it with 16S. So this is one of those instances where the best way to do it is not to come up with a fancier algorithm, but to improve your source data and create an auxiliary data structure and plug that in. But this resolves something that's been a long-standing problem in the field, and we've been applying it to different studies of autism, where different studies reach different conclusions because of these technical artifacts, not because the data was really different. And the really cool thing about this is that for the first time, we can see correlation in the effect size of different variables on the microbiome with these two sequencing technologies. So essentially what we can see is no matter which technology we used, we get the same size effect of the same variable, like how much white wine you drank, for example, which has a large effect, what population you're in this particular cohort, which has a smaller effect. And then things like, there's a lot of things that don't really matter for your microbiome, which have no effect in both cohorts. And I should say that this came from THDMI, which is a collaboration that we're doing with Danone through CMI. So part of the reason why this is important is that there's a lot of excitement in using machine learning technologies to do precise, personalized nutrition. And this paper from the Weizmann Institute came out at the end of 2015 and generated a lot of excitement because they showed that you could hook hundreds of people up to continuous glucose monitors, figure out the impact of each food on your blood sugar. And when you did the population average, it was exactly the same as published results. But if you did it person by person, it was totally different, one person to another. Then they showed that they could build a diet based on your individual responses to food and based on your microbiome that would either send your blood glucose spiking or keep it under control. And one of the big conclusions of this study was that the same food had totally different impacts on different people. So for most people, it's more healthy to eat rice, but for a significant subset, it's actually healthier for them to eat ice cream, at least in terms of their blood glucose. So this leads to that question, am I in the, how do I tell if I'm in the rice category or in the ice cream category, which is that your microbes decide that. But then the question is, suppose I find that I'm in the rice category, but I want to be in the ice cream category. Could I change my microbiome to get over there? And that might sound kind of silly, but it's the same question as for drugs, right? If I'm a non-responder for a drug, because of my microbiome, could I move over to become a responder for that drug by changing my microbiome? So we're working on this in the context of Alzheimer's on a large NIA-funded project, where what we're doing is we're piggybacking on a number of different studies that are doing dietary interventions and then reading out the microbiome and metabolome. This is another one of these large consortium projects. I'm not going to read everyone on this slide, but it's led here and Caltech and Duke University. And one of the signature achievements of the project so far has been better ways of reading out diet, not from the microbes, but from the metabolites they produce. And so this is the collaboration with Peter Dorrestein's lab here at UCSD, where we're doing a lot of multi-omic stuff together. And essentially, what we did in this paper was figured out how to take reference data from running thousands of foods through the mass spec and then match the molecules up to clinical samples by a technique called molecular networking that's implemented in GMPS. So the ground truthing was initially just to figure out, do fecal samples, are they different in their chemistry if they come from omnivores, shown in blue here, versus vegetarians, shown in brown, which they are.
And then a lot of the details make sense. Like if you're an omnivore, you eat more dairy and more meat. You eat fewer legumes and fewer fruits, for example. And so because it's an Alzheimer's project, a lot of what we were interested in was taking these food inferences and linking them up to memory. So using a score called Ray's auditory verbal learning test. And so basically for each food, we could take all the molecules derived from that food and then match it up to these scores. So, so the worst forgetting, it turned out, was from sugarcane and soda. And then the best improvement in learning was from things like fish, strawberries, and mushrooms. And you might think, so what? Is there any evidence for this besides that it came up in this data analysis? But when we went to the literature, what we found was that indeed there were a whole lot of press articles and a whole lot of studies on these specific things, linking soda and sugar to Alzheimer's. And then similarly looking at the decrease in Alzheimer's with fish intake, with strawberries and the anthocyanins they contain, and then by mushrooms. So what's amazing about this is that with a single assay doing untargeted metabolomics that you can basically do in less than a day in the lab, we're recapturing the results from literally decades of individual laborious research studies. And again, starting to fill out the database of what you would need in order to make these very precise dietary recommendations for individuals for specific phenotypes. And I should note that these microbiome-directed interventions are already saving lives in the context of cancer. So Jen Wago at MD Anderson has been looking at can you use dietary interventions like fiber and probiotics to take people who are not going to respond to checkpoint inhibitor therapy because of their microbiome and move them into the responder category. And what she's found is that you can do that with dietary fiber. However, probiotics don't work and actually kill you faster. And you can reproduce this in a mouse model where if you give the mouse a tumor and also the microbiome from the patient and then feed it probiotics, it will die faster reproducibly. So a lot of the stuff is counterintuitive and not what you would expect from the marketing of these products, which is why we really need more research in these areas. All right, so I'm just going to end by telling you a bit about what our vision for the future of all this is. And so this is really the science fiction part of the talk. But as I mentioned, the American Gut Project allows people to claim a pin for themselves on these kinds of microbiome maps, like the one defined by the Human Microbiome Project. Of course, not everyone wants to know what's in there. So these are middle schoolers touring our lab, learning exactly what we're going to do with their poop. But a lot of people have been fascinated. And the best reason why you want to know where you are on these maps comes from a disease called C. diff. And what we're looking at here is stool samples from patients with C. diff diarrhea, but you can see they look nothing like a healthy gut microbiome. And so what's going to happen is that 4 of those patients are going to get a fecal transplant. This is Bill Sanborn, who used to be our chair of GI, about to do his first fecal transplant with hospital-grade stool he got from a nonprofit called OpenBiome, because the FDA regulates stool as a drug for reasons that will become clear very soon. But you have to try to prove that you manufactured it like a drug, which is pretty hard if it's something someone literally pooped out rather than it was manufactured in the clean room, right? So what's going to happen, and this is a principal coordinates plot of unifrac distances using that HMP, Human Microbiome Project reference frame, and inserting a new data set into it. 4 of these patients are going to get a fecal transplant from that one healthy donor who was down at the bottom in that brown region, the healthy region defined by the HMP. And you might wonder, given that these are totally different habitats, are you even going to see anything on this whole microbiome scale transferring in a few grams of stool? And what we're going to do is we're going to animate the PKA plot over time. So each frame in this is 1 day in the lives of these patients who are having diarrheal episodes dozens of times a day to be eligible for this trial. And they've failed antibiotic treatment for 2 years. And I'm going to start this going, and what you can see is immediately, just 2 or 3 days, their microbiomes completely reset to the healthy state. And this is coupled to all of their clinical symptoms vanishing, and they're producing healthy poop for the first time in literally years. And you can see that they stay in this healthy region during the months of follow-up. So this just shows you the power of these kinds of visualizations. They're showing you what sick looks like, showing you what healthy looks like, and then showing you the rate and completeness and progress towards that healthy state. And so our overarching research program at the moment is to figure out what other diseases can you find these aberrant microbiomes for, and then figure out how can you bring them back into health, whether it's something like Parkinson's or obesity or autism or many other things that have been linked to the human microbiome now. So we need to find the good and the bad places on the map. But more than that, we need to develop a kind of microbiome GPS that tells you not just where am I right now, but where do I need to go? What do I need to do 10 by 10 to get there? Whether it's something that seems as extreme as stool transplant, whether it's better drugs or phage therapy, or whether it's something as gentle as diet, which there's no short-term fix for, but over the long term is one of the things that has the biggest impact on your microbiome and on your health. And so our dream with this is to develop this kind of system and make it so easy that even kids can use it. So when my daughter was 3, when we took this photo, she'd already figured out not just how to use the smartphone, but where we ought to frequently use them. And so the dream is that as you flush, maybe you can have the results immediately delivered to you in some sort of usable display. And back then, this was too slow to do on a phone, but now we can. And for example, this is the first 3 years of her microbiome data, starting off undifferentiated at birth, and then going over those first 2 years into the healthy states of the different body sites as defined by the American Gut Project. So it's the kind of thing where you could imagine that you could get very detailed guidance about are you going in the right direction or the wrong direction with respect to what you're eating, what your health conditions are, what your drugs are, by reading out this information that we're literally just flushing away at present. And you could imagine taking it a step further. So maybe you have a smart toilet that analyzes your microbiome every time you poop. But what we'd really like to do is to do it from molecules rather than from the DNA, and to do it from breath, which is where a lot of your microbial metabolism ends up. So the concept is that as you examine yourself in that mirror in the morning, maybe as you breathe on the mirror, your breath is whisked away into the back of it, into some sort of sensing technology, where you get some sort of display of the chemicals there. Unless you really loved organic chemistry, you probably don't want to see this first thing in the morning. So obviously working on user interface for that display is going to be important. But then the idea is that you could see the proportions of different kinds of microbes you have, like we do in the American Gut Project, and you could locate yourself on these kinds of microbiome maps, where if you're at risk for some particular medical condition, like inflammatory bowel disease, for example, maybe it would tell you to see a physician, or maybe it would tell you things that you can do yourself as a consumer that are evidence-based that would allow you to modify where you are on that microbiome map to a lower risk state. And while we're dreaming about this, you could imagine the mirror and the display on that mirror communicating back to your smartphone. So when you're faced with 1,000 yogurts in the supermarket, maybe you can scan the barcode of it and tell, does this product have the microbes or the molecules that I need to, in fact, shift my microbiome around on this map? But you could even take that a step further. You could imagine a version of ChatGPT, let's call it Crap GPT, where you can ask your phone, should I eat this particular product to improve my health, right? And have it do all of the chemistry and all of the microbiology for you. Or similarly, you might want to ask it, can you highlight on my phone with AR the products that I should eat instead of the ones that I shouldn't eat? And similarly, you can imagine scanning the QR codes of the menu at your restaurant and having it automatically do that processing for you, matching it up to your microbiome profile and where you want to be on that map to tell you, are you doing the right things for different dimensions like your blood sugar, your cognition, all of the other things that the microbiome is now known to affect. So what would we have to do to actually make this a reality? I think it's actually less hard than it might seem at first, right? So the first thing that we need is large cohort data on the individual response to different interventions, whether that's diet or drugs or exercise or other things that have high individual variability, where that individual variability is explained by the microbiome. And there's a lot of these efforts going on at the moment. The second is that we need better compositionally aware methods for analysis, and especially longitudinal analysis. And I dropped this in the interest of time, but especially multi-omics analysis, where you want to analyze several of those data layers together. We need better reference databases, and especially for things like the fungi, which are playing an increasing role in cancer and in a range of other diseases. We really need better ways. We need better complete genome resources and phylogenies that link them together, and better methods for integrating that data at the scale of millions or hundreds of millions of genomes, which is far beyond where those algorithms scale at the moment, even with XSEED resources, which we were already using to build those trees. And then finally, the microbiome and the metabolome are just two pieces of a much larger puzzle. We see a lot of opportunities for collaboration and integration with other AI technologies, especially things like large language models, especially things like image manipulation. So imagine if your mirror could show you what you're going to look like in 10, 20, 50 years if you eat the same way every day that you did today, or what you would look like if you did better or worse, right? So you could imagine gamifying that, changing the image in real time and showing people their score with respect to that image. And so there's a lot of things like that that we think have enormous potential to work together on to build these kinds of systems that are not just fun in the lab, but really have a real world impact. So with that, I'd like to thank the amazing people in my lab, literally over 1,000 collaborators whose work I mentioned briefly here. I apologize if I didn't mention you specifically, or if you sent me slides at the last minute and I didn't use them, or cut them out right before this. I'd also like to thank our many sources of funding, and in particular, NSF, who have supported a number of these graduate students on fellowships, and the Exceed resources and some of the mathematical work that I showed you with Manuel Yadza, as well as the Wolf family and the tens of thousands of members of the public who've contributed to the citizen science initiatives that we run. And with that, thanks again for the opportunity to speak here and to connect with you here. Be delighted to answer any questions that you have. Thank you.
Thank you, Rob, for that great talk. We have about 5 minutes for questions.
Thanks for the really interesting talk. I'm just curious, I mean, when you change this microbiome profile, how do you know the effect is local? I mean, like when you do this kind of transplant, what if there are other unwanted, unforeseen effects?
That is a great question that no one will fund research into. So unfortunately, the way medical research is organized in this country is that the NIH is divided into a lot of institutes and centers, each of which studies a particular disease. So the research on fecal transplant is essentially all funded by NIDDK, which is digestive disease. And in general, if you propose looking at the microbiome at other parts of the body, like the mouth or the skin or wherever, it just gets cut out of your research project and they do not fund that component. So that's really an opportunity for agencies that fund more basic science to get involved in finding out what are those long-range effects, what is the basic biology of the system, that kind of thing. There is some of that kind of integrated research going on in Scandinavia and in Australia and in Taiwan and Japan, but it's coming along relatively slowly, in part because they don't have access to some of the large-scale sequencing and compute technologies that we have for this stuff here. Oh, sorry, I think they want the microphone to Zoom.
Thank you for the talk. I was just curious to understand on a large scale basis, when could we see some of these technologies reaching the larger public?
Yeah, well, again, that's a great question. So there are commercially available microbiome tests now, but in general, they're proprietary solutions where you don't know what they do and you don't know what the weak spots are. So that's why we're in the phase of the technology development. We're having open science, full disclosure of all the protocols, full publication of all the data is still really helpful. And I feel like we're still in that pre-competitive phase where it's not necessarily ready to commercialize yet, but it will be fairly soon. Some of the companies that have commercialized, like Bioinformatics, for example, was valued at $500 million before it got raided by the FBI and shut down, and its co-founders are now fugitives in Europe. So there definitely are risks to trying to commercialize something that doesn't work yet, even if it's very hot, topic area. So in terms of clinically approved tests, so things like the cancer stuff that I showed you, there's a test for cardiovascular disease that basically tells you from the TMAO levels and your microbes whether you personally are at risk from eating red meat, or if you can eat as much of it as you like and it's not going to increase your risk. There's things like the Day2 technology. For full disclosure, I'm on their SAB, but that was commercialized out of the work from Eran Alanav's lab and Eran Segal's lab that I showed you on the diet prediction. They tried to do that direct to consumer, but that wasn't a very successful business model. So they're now trying to partner with insurers and large self-insuring organizations to do it at the population level, not the individual level. So there's pockets of it being available already, but in terms of large-scale adoption, it has to be cheaper. The results have to be more robust, and they have to be a lot more explainable. So the big problem with the kinds of classifiers that we're using at the moment, especially the ones that we get out of auto AI, is that we get a great classifier, but we have no idea how it works or why it works. And so XAI technologies are going to be really essential for turning it into something people can use. The other big barrier is auxiliary data layers. So you probably don't care about your microbes per se, but you care a lot about, can we link your microbes to health? And then the FDA cares a lot about what we say about how we can link your microbes to health, because if we're offering an unregistered medical test, then they get very upset about it. So, you know, understanding what claims you can and can't make, and what evidence you have to have, and what licensing and certification you have to make the claims that you'd really like to make. There's a lot of that kind of discussion going on. So at the Excite Lab, which is the COVID testing lab that we set up to process, if you've ever used the vending machines on campus, that's where those samples went, and we've done over 1 million tests there at this point. That whole process was fascinating because it showed the gap between what we can do for research, where we have the assay working in like 2 weeks in April of 2020, but then it took months and months and months to go through the FDA procedure to get it to where we were allowed to make it available to students and faculty and staff here, and do it as a clinical test that went into your EPIC record. But having gone through that experience, that gives us a lot of really valuable pointers to how we would do it for these more complex things in the microbiome.